Preparing. And now it is still setting up. Preparing. Here we go. Now we're live. Okay, what a morning. Welcome to Neuroccino, everyone. Great to see a full house today. The way it works is we meet every Monday morning, 9:30, and someone volunteers to present a paper. And today this someone is Anna, who's currently doing a PhD in Bordeaux. Anna, when you're ready, please share the link in the chat and take it away.
Thank you very much, Steph. We are going to share, so you should have the link now to a paper from Nature. Somebody is trying to enter our lab. No, we're presenting. You need to go. Okay, Michelle, I'll take care of it. Thank you. All right, and so today I'm going to talk about a paper that was just recently published on the 22nd of February, and it's about mapping dysfunctional circuits in the frontal cortex using deep brain stimulation. And they also managed to merge it with brain connectivity and like the connectome in general. So therefore, I thought it's pretty exciting since our labs are a little bit into brain connectivity, and I thought it's a good match, just a slight tiny bit. So I thought it's a good match. And there's also, so it's by Barbara Hollander, who is at the Charité, and a bunch of other collaborators, and also Andreas Horn, who Marcella is going to visit in Harvard this fall. So I thought maybe a cool paper to present. So let me share my screen first so I can show you all the nice figures. I hope this is the correct screen. Yeah, that's it, right? You can see everything? Awesome. Yes. All right, so I'm hearing myself double because Michelle is not here, and now he's back.
We hear you okay. If it's not too irritating for you, you can just carry on.
Right, so so just to give you a general introduction and what was behind the study. So they studied especially frontal circuits that are crucial in for like motor, cognitive, and affective processing. If those circuits are disrupted and have dysfunctions, that is associated with various brain disorders. And here they focused on 4 brain disorders, namely oh my gosh, I already forgot. Namely, Parkinson's disease, dystonia, Tourette syndrome, and OCD. So what they basically studied was then the relationship, the relationship between specific frontal domains and then their dysfunctions. And for that, they examined 534 electrodes for deep brain stimulation, and tried to identify how basically deep brain stimulation modulates connections for optimal therapeutic response for these 4 disorders that I just named. So so what they, I'm sorry, I have all my notes here, old school on paper still, but what they did was stimulating the subthalamic nucleus. And there is a paradoxon they basically explained that, so that usually there is, hold on. So these interconnections cover motor, cognitive, and affective functions, and are implemented as frontal subcortical circuits that cross-communicate but retain a certain degree of segregation on thalamic level. And so there is the primary input structure, which is the striatum, but the subthalamic nucleus has been also recently identified and recognized as the second direct input nucleus. And since, it's much more smaller, but here they managed to explain why they stimulated the STN by having it basically as a node to distinguish the circuits in the 4 different brain dysfunctions. So Parkinson's, TS, dystonia, and OCD. So let me walk you through the pipeline and how they managed to do it. Let's take a look at this figure here. So you can see it, yeah. Okay, so the first panel here. Yeah, so this was the first thing that they wanted to do, is find a sweet spot within the STN for deep brain stimulation, right? So for that, they first, for each patient, the electrodes that were implanted for deep brain stimulation were reconstructed to basically determine the specific position within the STN. And from there, they generated this E-fields in standard space. So basically, they looked at the electric field magnitudes in those patients that were generated by the deep brain stimulation, and then they tried to map that into the standard space in order to then, as you can see here, calculate this voxel-wise Spearman correlation between the E-field magnitudes that are associated with clinical improvements, contrasted to the E-field magnitudes and their positions, basically, that were not associated or associated with poor improvement. And from there, they generated this sweet spot profile, as they call it. So we have, obviously, they did that separately for each disorder. So for dystonia, for Tourette, for Parkinson's, and OCD. So you have those different sweet spots within the STN that are, they call it the segregation in neuroanatomy dysfunction mapping. So for the target level. So you have within the very, very small area, the STN, different sweet spots. They also have sour spots, but those are not, those are not pictured here. And then the second chunk of their pipeline was the DBS fiber filtering, which basically involves the normative connectome. So they took over 900 participants from the HCP, and I think there were more than 6 million fibers, basically, or streamlines, that they then, one for one, tried to, so they put the, they applied basically voxel-wise correlation again across the entire brain volume. So yeah, with each streamline. So basically, each streamline that was in a predefined normative connectome here was analyzed for its relevance in distinguishing good or positive clinical outcome from bad or for poor responders of each disorder cohort. And by that, they looked at the R value of each streamline. So here, for example, this example streamline was predominantly modulated by top responders because it's like completely red, so the R value is high. And then they created those discriminative streamline profiles for each disorder, which they called the sweet streamlines. So they also contrasted basically for each disorder the streamlines, and by thus then in a bigger picture the circuits that are involved into this specific condition. All right, so until now clear? Yeah. Okay, cool. Let me go back to the paper. There we go. Okay. So there, okay, here we have the results. Oops. Oh, so here you can also, again, here you can see the streamlines basically, and how they were, how the electrode placements were relative to the SDN across the discovery cohorts, like across the 4 different conditions. And we also have a slight difference in the sample sizes. So for dystonia, we had 56 patients, for Parkinson's 94, for Tourette 14, and OCD 19. And the thing is, the people validated everything also on external cohorts, but of course, the models that they created for example, the Tourette syndrome, where you have the sample size of 14, they weren't as stable as the ones, for example, for Parkinson's. So it's, sample size was also a little bit of a problem. Here, okay, we can skip that because this is basically what I just showed. It's just a segregation of the dysfunction mappings on subthalamic level by, again, disease specifics, but here they topographically, map that. And this is just a little bit more in detail, but looking at the key findings, or at what they actually, concluded from it. So by applying this pipeline and basically merging the sweet spot mapping from deep brain stimulation in the STN and fiber filtering afterwards, they managed to identify basically specific dysfunctional circuits within the frontal cortex that are related to all these 4 disorders. So that you can, see here. In dystonia, it was mainly the sensorimotor cortices and like their connections. Then in Tourette syndrome, you can see the primary motor cortex, for example. In Parkinson's, you can spot, probably Stephanie can spot it better because she's the anatomy expert here, but just reading what they concluded from it. In Parkinson's, we have the supplementary motor area, and in OCD, the ventromedial, prefrontal, and anterior cingulate cortices. So this basically, they managed to demonstrate a topographical arrangement of dysfunctional circuits, basically from even, occipital, frontal, from occipital to the frontal regions, and you can spot it here. This is maybe this is even a little bit easier to read. So we have, maybe I can make it a little bit smaller so we can see the legend. But here you see again color-coded what the legend here. All circuits basically plotted into the brain, and these are basically associated with therapeutic stimulation effects that are affect the dysfunctions in the brain for those 4 different disorders. All right, so they also, for like, obviously there were limitations combining all these, like a lot of different cohorts from a lot of different centers, but they did a bunch of work that I'm not gonna mention in detail to normalize everything and to control for as much as possible, to validate it externally. And what I found, so what I also found really interesting was that they actually applied it on 3 patients. And I'm gonna try to find it here. There we go, I think, the second programming. Okay, so for example, I'm just gonna read that out because, so by identifying special locations where you can stimulate such a small brain structure, which is involved into symptom release in, for example, OCD, right? They managed to do this in 3 patients because the data that they used, it's one of the limitations as well, it was done on retrospective data. So of course we need some more proof, some more validation, which is also something for the future, I think, to maybe look into how that would match with the functionectum, for example. But I'll just gonna try to, I'll just read that out here. So there was a reprogramming patient, a 20-year-old female with OCD, and they implanted a conventional omnidirectional lead targeting the region that they have basically identified, which was important for symptom relief in OCD. And that led to an improvement of 6 points on a scale that is basically that measures OCD from 35 points to 29. And 1 month after that, so that was, I think, on the first day or in the first couple of days, and 1 month after, they call it the streamline-based reprogramming, this score reduced to 22 points. So this is pretty impressive. There are a lot of people coming into the lab today. So this was pretty impressive that they were able to show it actually on real patients. And to conclude, okay, this is also a little bit more into detail. There we have the discussion. So they had 11 patient cohorts, pretty large sample size for that. But to conclude, so this study basically showcases network effects of intracranial brain stimulation, and they show basically that this can be a tool that can be used for, first of all, investigating the circuits, the cortical circuits, and clinical dysfunctions. And they started to map they call it the human dysfunctum. It's also, they demonstrated a topographical organization of those dysfunction mappings across neuroanatomical levels, and provided insight in the interconnectedness of the prefrontal subthalamic loops, they say. And also other subthalamic subterritories, but that's a very specific area, but that can be obviously extended to more, for example, by looking into other clinical dysfunctions, or even looking at not even, brain disorders, but just at functions, for example, language. That could be also interesting to see whether something can be done there. This is pretty significant since they identified basically circuits for therapeutic targets, and were able to showcase that as well. This can be also, I guess, used at some point if this functum gets a little bit more evolved and developed. This can be probably used as something in the clinic that would support decision making and obviously treatment. I think let me maybe just mention a couple of limitations that they were also talking about, if I find them. So one of the limitations that I talked about earlier was that they mainly relied this whole analysis on retrospective data, which could have led to bias in, for example, interpreting the clinical outcomes. And there is also, there are challenges basically in accurately capturing this physiological effects that are underlying with the brain dysfunctions with a very simplified model that is used for basically tissue activation. There can also, so you saw in the pipeline in the beginning, I'll try to actually, it's interesting to just open the pipeline. I think it's the most, it's a impressive work that they put together here. So here, for example, they try to map it all on a 3D voxelized grid, but of course there that could also have led to mismatches and warping those localizations and putting them into a template space. Although they try to use all the other pre-processing pipelines to control for it. And I think the last thing is that they also mention is that they need further validation and for the findings and also for the reported streamlines in not only a normative connectome as here, but maybe also in patient-specific data. So before this can actually go to a clinical application, but I think I'm running out of time here, so I'm sorry. I'm done. I hope that was clear. It's a super interesting study, but a little bit stressful today with so many visitors in the lab.
Oh, but it's all your friends. You know everyone in the room. Thanks for presenting the paper. I've seen it come my way over and over again over the last couple of days. So I'm very glad that you chose it to present it, and I think you did quite a good job given the complexity of the paper. Now, are there questions? We have a few minutes.
Michelle had one in the chat now. What about the false negative? You mean the false negative in
Wait, what?
No?
Yeah, so with spectrography, you don't see the lateral fanning of the connection. So I thought actually in this specific case, when you look at the connectivity of small region of subcortical nuclei, functional connectivity is better than structural connectivity.
But
that was more a comment than a question about.
Right, but from functional connectivity, you can't make causal implications. This is just correlation, no? And from deep brain stimulation, you can actually, well, if
I mean, they could, instead of illustrating the sweet spot connectivity using structural connectivity, they could have used functional connectivity. Unless they can stimulate directly the white matter pathway and outside of the subthalamic use.
Do you wanna switch microphones and you elaborate a little bit on that, Mish?
Yeah, I've been in and out. Entire presentation. Sorry.
Much better.
I've been in and out the entire presentation for technical reasons, but so my can you turn off?
But switch off your volume.
I'm not. Thank you. So my understanding is they show the connectivity of the sweet spot of the subthalamic nucleus using structural connectivity. So looking at streamline, diffusion weighted imaging. That is actually not so good to show projection pathways because we don't see the fanning onto the entire cortex. So I thought actually in this present case, because this is a team that is really expert in functional connectivity, I'm glad they use structural connectivity, but functional connectivity would have been more appropriate if they wanted to highlight the areas and the set of areas that are connected to the subthalamic nucleus. But that was more comments than the questions, but I'm happy to
But
hear.
with structural connectivity, if you use the right algorithm, you can reconstruct the lateral projections slightly better than before. So why do you think this is suboptimal for this particular study? Now you're both mute. You guys, you got to work out your setup in the office.
Okay.
Nope.
What did you say, Steph? Sorry.
Right. The question was, you talked about the lateral projections not being there in structural connectivity, but that partially depends on the algorithm that you use, how well you can reconstruct them. So why do you think that this is not good enough in this case?
So, if you go back onto like the result of the structural connectivity, which is like figure 2 or 3. Can you go on figure 2 or 3, please, Anna? Sorry.
Oh, this is 2.
It's 3 then. Well, maybe figure 4. So it was maybe figure 4.
Here we go. Here we go.
Yeah. So you can see how your tractography is stuck onto the midline or mid portion of the brain, and we do know the subthalamic nucleus from axonal tracing studies also project laterally.
It definitely looks suggestive that you only reconstruct the medial aspect, but it's hard to definitively judge that on a sagittal section. You got to give them that.
Right, but I've been playing.
I totally see where you're
HCP.
coming from. It does look like we're missing the lateral projections here.
But if you go in figure 5, then you have the projections.
Right.
And you can see the typical pattern of what you can see with structural connectivity, which is limited due to the crossing with other fibers.
Yeah.
Okay. It's great study. I didn't want to say that it wasn't good. I just said maybe functional connectivity would have given like a more comprehensive vision of the cortical areas.
Well, so you don't have to do one or the other, right? A combined approach would have also been a nice way of going.
That's right. That's right.
Also, can you mute yourself? Next time I have headphones again. Also, they had a very, very large data set of, I think, 16 institutes, and having that data, like functional data for all of them, all of the patients, it might have been maybe, way more complicated to get.
True, but you could make the same argument for the high resolution structural data in the end, because it's not a common sequence that you get on a clinical scanner anyways. But the good thing is you can ask them when you're there. Right. Us discussing killed one of the questions, but one hand is still up. Mugge.
Oh. Hi, yeah, thanks, Anna, for presenting this paper. So I was just looking into the paper, but I couldn't figure it out. So they have these sweet streamlines, but do they like discuss where they originate from? For example, the sweet streamlines for the Parkinson's disease. So because in the STM there are these like different subsections, the motor area, limbic area, or the like associative area. So do they discuss like whether these different streamlines, whether they originate from the different subsections or the same subsection? I couldn't find anything about that part, but maybe I missed.
I'm not sure if they discuss where they originate from, but they use the STN as a node. Like they use that as to basically say that's where it's where they all meet, and by contrasting the sweet where am I? The sweet streamlines, they were able to identify like sweet streamlines for every disease separately, although only the SDN as such a small structure was stimulated. So I'm not sure. I don't think they're talking about the origin. But I might, yeah.
I'm just confused about like they have these DBS outcomes from these streamline-based reprogramming that they call. But what are they? Is there any other comparison? Like what is the they say that the outcomes are improved, but with respect to what? Like what were their original reprogramming? Like I thought it would be the different like SDN sections, but okay, maybe.
It's the clinical, so they looked at, so it's about the improvement in the clinical symptoms, right? So that
so
you
they're
can
not okay.
Here, so yeah, percentage of clinical improvement following the deep brain stimulation. So that is basically what they are looking at.
Okay. Okay, thank you. Thanks.
Sorry if I couldn't fully answer your
this
question.
is very interesting. It's just, yeah, I'll read this in more detail for sure. Thanks.
Final question, Eduardo.
Yeah, just a quick update on what Michelle was saying. Maybe they cannot collect functional data because I don't know, Hannah. Maybe you can tell us more about that, but I don't think those patients can undergo any kind of fMRI scan with the electrodes in place. So
They gotta sort out their microphone situation first. Mish wants to talk.
So in the present case, the tractography they use is a tractography from the Human Connectome project, which means they use the localization, the sweet spot, and track in the Human Connectome project. And the team of Aaron Bose and Michael Fogt, who's not a co-author here, they're really good in doing the same thing, but with functional connectivity. So they use resting state from healthy controls, and they use those small region as seed in this database of healthy control. So there is no need to do resting state functional MRI in the patients in order to assess the connective properties of the sweet spot. So that's why I was surprised not to see them like using it in complementary to look at whether there was similarity or differences with the current. Yeah, somebody said something. No, okay.
yeah, okay, thanks for clarification.
Right, that's all we have time for today. Thank you very much for presenting the paper. Clearly, we all have to read it and dive a bit deeper into it, but very exciting. Thank you. Have a good week in science, and we shall see you next Monday. Bye.